I remind you, the younger participants, if you are not vaccinated, it's Friday, so please do a test in the coffee break. I, for example, I'm vaccinated, but I tested myself 2 times a week because I was coughing a little bit. So also, if you want to be sure, if you are vaccinated, of course, you can take the test. We have plenty of, and also if you need one for a family member, also feel free to take one. So now I welcome our next speaker. We were expecting him to be here in person. Unfortunately, due to some other tasks, it was not possible, but due to our good tech support here, we have him now on the screen, and we will also stream him. So welcome, Professor Alexander Scholos. And he is giving a talk about nanostructured materials and in connection with patch clamping. So, please, Alexander, introduce briefly yourself. He is on the phone. Oh, yes, it's this phone. Maybe they are still solving some problem. Oh no, it looks nice. Okay. Okay, let's give them another minute. He has also been asked to shorten his presentation that the coffee break is a bit longer. As you see, we are a bit behind the schedule. This was because we have been waiting a bit for you, and those who dropped in during the lecture missed a very nice intro. Actually, we didn't know that our previous speaker, Yevgen, is a brilliant piano player. So he gave a private piano concert for the lecturers and for one participant until more participants dropped in. When we made the test.
Кружочок тягніть вправо. Цей, так, кружочок вправо тягніть.
We were already talking to him this morning.
Та нічого, не відчую, тягніть кружочок.
Hat Probleme.
Окей, ich kann schneller. Я ж не знаю. Я можу швиденько вам Google Meet створити. Добре. У вас настройка треба в ноутбуці додати звук.
Okay, try out.
Якщо не так, не чую. А я ж... О. Значить, чекайте. А ну, боже, звідки я бачу? Краще.
Yeah, unfortunately, our support is
Тут. Це точно той аудіовихід? Слика. Може сюди? Все, я виключила.
That sounds good.
Нормально?
Так, все на ващу. Все на ващу, як і бачу.
Презентацію зараз видно?
Так.
Good morning, everyone. First of all, I would like to thank the organizers of this great event in Ukraine and for the opportunity to contribute to this event with my lecture. Apologies, I cannot do this offline because we had a sudden change in the time that my regular class is given. I'm at the Institute of Biology and Medicine now. But I hope you can hear me okay and you can see my slides. Is everything okay?
Yeah.
Okay, great. Thanks. So my talk will be about nanostructured materials and how this can be used to modulate activity of various ion channels. Basically, I would like to pose this big question: how compatible are they? I mean, nanostructured materials and nano-sized molecules. In this case, ion channels, and we use different technical approaches to address this question, from molecular modeling to technical investigation of these interactions. So, in this lecture, I would like to first to say a little bit about ion channels as very important class of biological macromolecules, introduction to ion channels as light resistors. Then I think this part is probably almost obsolete at the end of this talk. You have heard already so much about various nanostructured materials. Still, I'd like to show a few studies done in this area before we commence our work. And then these last 5 parts will be about our own work on various nanostructured materials, and just recalling the excellent talk from Professor Stoika yesterday about smart nanomaterials. I also included a brief section on liposomal quercetin as another nano-sized preparation. Okay, so coming back to that big question, are they really compatible, these structures? So these are just some illustrations, and we work a lot on carbon C60 tolerance. This is collaboration with Professor Yuri Prilutsky, who is my good colleague at the Department of Biophysical and Medical Informatics, and also this is a collaboration with his colleagues in Germany at the Technical University of Ilmenau. Now, talking about ion channels, this is just a cartoon, very schematic representation of nicotinic acetylcholine receptor. So maybe some of you, if you don't know what ion channels are or what they do, this is, I would like to mention a few basic points. So these are typically multimeric complexes. In this case, we have 2 alpha subunits, beta, gamma, and delta subunits. And you can see the channel pore is closed after acetylcholine binding to molecules of acetylcholine bonds. The channel pore is opened. And ion channels are generally classified in 2 large groups or classes: voltage-gated channels, depicted here, and receptor-operated channels. And we do lots of work at the single channel level. Here you can see transitions between closed and open conformation of the channel. And that's the beauty of preclinical techniques that allows us to look at the mechanism of drug action. So we can see whether a drug alters single channel conductance by changing these unitary current amplitudes, or maybe a drug can alter durations of open and closed states. So it's all doable with magical techniques that we use a lot in this research. So now a few words about the importance of these molecules. At the moment we know more than 340 genes encoding ion channels, which are present in our genome. They show extremely fast transitions, conformational changes between closed and open states in less than 10 microseconds. They ensure a very high rate of ion transport, kind of 10 million ions per second, which is comparable to simple diffusion in water. Channel pore, which is important in the context of our molecular modeling, including molecular modeling, is rather small in sub-nanometer size. For potassium channels, for example, approximately 0.3 nanometers. We currently know more than 60 human diseases, and in case of ion channels, this group of problems, clinical problems, got a name, special name, gemellopathies. And some of the most important ones are cystic fibrosis, disturbance of heart rate, cardiac arrhythmia, pain, chronic pain, and cancer. So these molecules are important targets for various drugs. So this is just an illustration of our early work on the menthol and cold receptor tryptamate. Illustration of the patch clamp technique is here. So we can electrically isolate one single channel in a small membrane patch, and then we can record these transitions. Here it's closed information and channel, you can see it opens very briefly for various directions. And we have done molecular modeling of tryptamate, as shown here on the left-hand side, using template of TRPV1, the capsaicin and heat receptor. And according to this model, especially we paid lots of attention to the selectivity core, so you can appreciate from this molecular modeling part that the core is kind of breathing. It's here it is in its closed conformation, here's the baseline, intermediate, the channel starts to open, and here is fully open channel, which corresponds to ion flow here. So, as I mentioned, there are more than 340 ion channels known, and this is a nice illustration of the genetic relations between them. And in case of voltage-gated ion channels, the diagram is shown here on the right-hand side. And in the literature, sometimes you can see this colloquial reference to ion channels as light transistors. They're really very important for generating electrical activity of different types of cells. And to do that, voltage-gated ion channels, they have transmembrane domains that carry positively charged amino acid residues, which are sensitive to magnetic potential. So, in this case, potassium channel open probability is illustrated as a function of magnetic potential, quite similar to what we see in physics of transistors. Okay, talking about practical importance of these molecules, there has been a recent survey published in Nature Reviews Drug Discovery in 2017 of molecular targets of FDA-approved drugs. And that was the great news for people working in the area of ion channels like myself, because if you look the number of drugs is listed in this table, but it's more revealing to look at these two diagrams. So, proportion of human protein drug targets in major families, in the pharmaceutical industry, these are known as privileged protein classes. So here we have a significant dominance of ion channels, 19%, as opposed to 12% of G protein coupled receptors and other protein classes. But now, if we look at what happens in this category, FDA-approved drugs, here we see the opposite. 33% of current drugs are acting on G protein coupled receptors, and approximately on 50%, twice as less, 18% are acting on ion channels. So there is obviously an important discrepancy, and we obviously need more molecules working as ion channel modulators. And this is what our research is about, to find such molecules, including nanostructured materials, which is the topic of my lecture today. And this is just another diagram from the same paper, illustrating physiological systems and pathological conditions where these molecules are so important. So here, this is according to international classification of disease states. For example, N means nervous diseases, So you can appreciate, especially these are especially important for the nervous system, quite important for sensory systems and respiratory system, and also very important drug targets for the treatment of cardiovascular diseases, as shown here. Now, I hope I'm kind of sort of convinced you that ion channels deserve a closer look. And talking about channelopathies, it's quite difficult for me, of course, to illustrate problems like cancer development or maybe cardiac arrhythmia or migraine or chronic pain, for example. But some problems are very much obvious if you look at skeletal muscle function. And here, this is illustration of myotonia congenita. Luckily, quite rare condition, kind of muscle stiffness, muscle spasm. But here I would like to show you a brief video from National Geographic. And this animal is known as myotonic goat, and you will easily see a problem with this animal. It's called also nervous goat, stiff-neck goat. So it's a temporal stiffness of legs, so it cannot run any longer. And the reason for this is that instead of just temporal action potential discharge in normal animals, these animals, they have continued action potential discharge after stimulation due to dysfunctional chloride channel in skeletal muscles. And another good example is this horse. These horses are called, this is a special breed, quarter horses. So you can notice here lots of spontaneous skeletal muscle contractions happen in these horses. But obviously they look very athletic and they are used in sport, actually, these horses. And in this case, it is a dysfunction of voltage-gated sodium channel. And finally, one last illustration of potential problems is this one. It's about peripheral nerve hyperexcitability due to dysfunctional voltage-gated KB 7.2 channel. And you can see manifestations of this problem. It's just kind of uncontrolled involuntary contractions again of skeletal muscles. But in this case, due to spontaneous action potential discharge in peripheral nerves. And if we can have a quick look at the structure of this channel, because I will talk, that's a gene and, sorry, I'll show you structure separately a little bit later. Now, but historically, channelopathies or some obvious phenotypes have been very instrumental in discovering new ion channels. And in particular, I would like to mention Drosophila TRP phenotype, because I will talk about some TRP channels that we study. So these are electroretinograms responses. So you can see during light stimulus in healthy flies, the response is more or less sustained, but in Drosophila TRP mutant flies, this response is transient. That's why these flies, they are blinded by light stimulus, especially bright light. They become blind and they can be identified very easily. And from this Drosophila, mutant Drosophila, scientists learned that there is an important ion channel involved in phototransduction, and it was named TRP, or transient receptor potential channel. And currently, we know that in mammals there are 28 members of these such proteins. They have been identified by structural homology, and they fall into 6 subfamilies. And they are very important sensors of various stimuli. For example, here TRPV1, the heat and capsaicin receptor, TRPM8, the cold and menthol receptor. I will talk today about TRPC4, which is receptor moderated channel in a different class. There are also stretch-activated and chemisensitive channels, pH-sensitive, those which sense oxidative stress, and so on and so on. An almost endless list of various sensory functions. And especially, I would like to mention today thermosensitive TRP channels. You see there are at least 6 of these. They have very high Q10 or temperature coefficients in excess of 5. At least some have Q10s of 20 and even more. These can be activated by changes in temperature according to this scale, and you see each of these is responsible for detecting a specific range of temperatures. But they can also be activated by natural products like menthol, capsaicin, I already mentioned, and some other compounds. And the reason I'm mentioning these today, you probably know that on the 4th of October, the Nobel Prize in Physiology or Medicine for this year was awarded to two American scientists, David Julius and African. And interestingly, this is what was said about this discovery. This is basically about our ability to sense various physical stimuli like heat, cold, and touch. And these are some key papers they published while discovering the capsaicin receptor, TRPV1, the cold receptor, TRPM8, and touch receptor, actually 2 proteins, piezo1 and piezo2. So, with that, let me just move to our next part of this discussion, and let's talk about nanostructured materials as possible modulators of ion channel activity. A lot was said already about nanostructured materials at this school, but I would like to quote also Nobel Prize winner, American physicist who worked in the area of quantum mechanics, who said at the atomic level we have new kinds of forces and new kinds of possibilities, new kinds of effects. So basically our research on nanostructured materials as pharmacological modulators of ion channels it's probably very well reflects on this expectation of new possibilities. And also you see some molecular modeling studies and analysis of sizes of these molecules suggested quite long ago, 2013, with this review, it was already highlighted that nanoparticles can be very well compatible with biological nanomachinery, including actually membranes and ion channels as well. Here, continuing from that paper, size-wise, most of biological assemblies of molecules and their complexes are within the nanometer range, similar to most common types of nanoparticles. And among possible targets, you see ion channels here are listed as well in that review. For my talk today, the most relevant ones are full-length C60 and single-bond carbon nanotubes. But there are, of course, many different types of carbon nanostructured materials. Some early studies have been done using molecular modeling, and they actually suggested that such interactions are quite possible. And please especially pay attention that in this video, ion channels have been highlighted already as potential drug targets for the action of nanomaterials. And this is the structure of potassium channel. It is composed of 4 subunits. It's a tetramer with a pore region through which usually potassium flows, but in this case, maybe carbon nanotubes and fluorines may occlude this channel according to these such molecular dynamics, molecular modeling simulations. Also, there are various biomedical applications for C60 fluorines. I'm sure well, you know this well even from this polygon. This was mentioned many times. Especially they are proposed to be used for drug delivery and things like that. They have antiviral properties and many other interesting properties. But there is, of course, this big question about the possible toxicity. And what I will show you in terms of blockade of ion channels, that probably reflects on possible biomedical application of these nanostructured materials, but also raises important questions about possible side effects. So, one point with these slides, keep in mind that so we need some balanced view here. Not get too much excited about benefits, but keep in mind the possible toxic effects can also occur. Now, molecular modeling of C60 interaction with potassium channels was done in 2010. And here, this is the trajectory of fluorine as it moves before occluding potassium channel core, which is depicted here. These are amino acid residues shown with red hands, which allow selectivity of this structure for potassium. And these are aggregates of fluorines. They can also interact with protein molecule forming ion channels. And there are also molecular modeling predictions that C60 fluorines can also bind to voltage sensing domain of voltage gated potassium channels as shown here. And just to continue here as well. In this case, this is voltage gated KV 1.2 channel. Now, that study also concluded that looking at the energetics of these interactions, this is also quite possible. So depending on the structure and properties of our ion channels, various possibilities exist. And they evaluated energetics of binding to the selectivity filters, to the external part of the channel selectivity filter, transmagnetic domain, as well, and also intracellular parts of the channel. So you see here somewhat different energies of binding, but they are in about the same range. And here they again raised that important question about possible toxicity of such molecules. Just to conclude previous molecular modeling work, this is a very nice illustration of using classical molecular dynamics, how C60 fluorine can geometrically clog passage of potassium ions through voltage-gated potassium channel. Now, molecular modeling is of course great, but what about experimental evidence? Do we have a good experimental evidence that such interactions are possible, first possible, and second, biologically relevant? I mean that these interactions really change ion channel activity. And there are not many papers in this area. The first one was published in the Journal of Biological Chemistry in 2003, where single-walled carbon nanotubes were posed as a new class of ion channel blockers. And these are transmission electron microscopy images showing the sizes of these molecules. And one conclusion from this paper is that diameter of these structures was a determining factor in these interactions. And then actually they also looked at C60 fullerenes with the same paper, but didn't mention those in the title of the paper. But now, if you look at the experimental evidence, you can appreciate that these effects are actually quite numerous. They expressed different types of potassium channels in Hector 3 or CHO cells, and they looked at the effects of single-wall carbon nanotubes and in this case fullerenes as well. And you see in all cases, these are very structurally very different channels. In all cases, there is quite obvious inhibition control in the presence of single-wall carbon nanotubes. And here, again, quite substantial inhibition. But there is some degree of selectivity as well in case of chloride channels mediated by CLC3 proteins. There was practically no effect of single-wall carbon nanotubes. What is of concern, of course, single-wall carbon nanotubes inhibited HERS channel, and this channel is very important for cardiac electrical activity. In fact, this is rapidly rectified channel which determines duration of action potential in the heart. And I should say here that a lot, this is very, it is so important for electrical activity of the heart that legislation is such that if any drug inhibits this channel, then it cannot be further developed to become a medicine. And here you can see quite a lot of inhibition. Also concentration dependence, of course, it's a big question. At low concentrations, there was actually no effect, but at higher doses, there was inhibition of this important cardiac channel. Now, with that introduction, I would like to move to our own work in this area. I will try to catch up in terms of time to let you enjoy your coffee break on time. So maybe I will go through some slides a bit quicker, but I will leave some time for questions just in case. So I would like to start with, as I mentioned, you've heard already the great talk from Professor Stoika about smart nanomedicines. So we worked on liposomal drugs as well, and we developed liposomal quercetin and novel nitric oxide donor and tested them and showed that they are very efficient openers of large conductance calcium-dependent potassium channels, which are denoted like this. B means big conductance, K potassium, and calcium-dependent channels. These molecules were developed in collaboration with our colleagues at the Institute of Pharmacology and Toxicology of the National Academy of Medical Sciences in Ukraine, and also some chemists working in Kharkiv. So just to introduce you to this work, I need to mention, to say a few words about structure of these channels. So it also, like TRP channels, it also has 6 transmembrane domains, and it's a tetramer. Here, 4 subunits are shown, and there is a central core, which is actually very small. I wonder if you can see the tiny white dot in the here, that's the channel core. And all this huge protein machinery is meant for calcium and voltage-dependent regulation of this small core. And these are domains involved in calcium binding, shown here, and voltage sensitivity in the S4 transmembrane domain. So actually, you can appreciate a simple fact from such images. The ion channels are not simply core in the membrane, but they are physiologically regulated pores, which open and close according to physiological needs of a cell. Now, what we've done in this area, these are empty liposomes, and liposomes incorporated quercetin. Quercetin is a flavonoid of plant origin, and many molecules of plant origin. They are actually hydrophobic, and it's a problem for delivering such molecules to their targets because of this hydrophobicity. So we thought it would be a good idea to include them in liposomes and deliver them as a complex. So what we found, we did this work at the single channel level, which I already explained. And interestingly, you can appreciate even from this one slide how this may be a very good idea to include a drafting liposomes because it enhances its bioavailability, makes it much more potent, and hopefully it also makes it, in terms of pharmacokinetics, a better drug. Here you can appreciate these differences. When we apply quercetin alone, there is increase in the frequency of channel openings. Obviously, that is controlled, and this is with quercetin. Illustrated some staged areas are illustrated here, and here we plot open probability, which is a fraction of time, total time the channel spends in its open conformation. Okay, so open probability of being calcium channel is increased by quercetin, but not actually very much, maybe twofold, depends. Now, empty liposomes, interestingly, also have some effects. And that is understandable because proteins are incorporated in lipid membranes, and adding another lipid to the membrane environment can alter channel properties. So there was also some increase in channel open probability with phosphatidylcholine in this case. But now, if we apply quercetin in its liposomal form, we have really huge effect, increase in channel open probability illustrated here. And here, suddenly we see that in a small membrane page, we have up to 7 active proteins. And this is an illustration, summary of the effects on channel open probability, or in other ways. In other words, this is a nice biophysical parameter to quantify the effect of a drug at a single molecule level. Right. Also, what is important in talking about liposomal quercetin, what we found that it can repair channels after they have been damaged through exposure to oxidative stress. Here we used hydrogen peroxide, H2O2, and these are current voltage relationships in control. Basically, current amplitude plotted as a function of voltage. In control after hydrogen peroxide application, and then still in the presence of hydrogen peroxide, channel activity can be restored by liposomal quercetin. And lots of diseases have this component of oxidative stress, so I think this is quite interesting finding. Now, another drug which we developed again with the Institute of Pharmacology and Toxicology, it's a liposomal donor of nitric oxide. It's a complex of liposome, cytochrome C, and nitric oxide included in phosphatidylcholine and dipalmethylphosphatidylglycerol liposomes, which have size from 90 to 100 nanometers. And we showed that both at the whole cell level, whole cell currents can be enhanced by this donor of nitric oxide quite substantially in a concentration-dependent manner. And associated with this increase, there was a negative shift of the activation curve, which is kind of biophysical mechanism of this effect. And here again, we can see at the single channel level what happens. Single channel openings upwards in control, and then with increasing concentration of liposomal and or donor, we can see that the activation can be really massive, shown here as open probability, depending on concentration of liposomes. And this work at the single channel level, it's actually it allows us to look at the mechanism of gut action, which is obvious benefit because if you look at the level of cells or tissues, sometimes it's not easy to know what really happens when we apply that. But when we work with one single molecule, the answers become quite easily obtainable. So here, illustration, you can see illustration of channel gating in control. That is big calcium channel. Occasional openings, not much. With liposomal nitric oxide donor applied at 10 micromolar, we have very substantial activation of the channel, so the channel opens very frequently. And these are amplitude histograms showing what happens here. In short, single channel amplitude didn't change. That means this nitric oxide does not affect channel permission. What it does instead, it alters the ratio between mean closed and mean open times. Mean open times are actually not very much affected. They are increased about twofold. It's not such a big deal. But mean closed time is decreased very substantially, and these are just the ratio between these. So the main effect is to induce frequent channel openings because closed conformation of the channel becomes destabilized. It's not stable any longer. The preferred state becomes open state of the channel. Okay, moving forward. Now let's talk about materials which are really of nano-sizes. And I would like to start with nano gold particles. And these were developed again with the Institute of Pharmacology and Toxicology and also with a commercial company, Nanomedtech, which is in. So they developed plasmonic nano gold particles, and we found one very extremely interesting effect for us because these molecules can work as remote switches of channel activity. We can control this effect using laser. I actually, at the beginning of this study, I could not believe this would be possible because I know one paradigm of pharmacology is that a drug should bind to its target to produce a pharmacological effect. In this case, we have an effect which can be induced in a remote manner, and moreover, you can switch it on and off easily using laser illumination. And this is because it is based on plasmonic resonance. So this is an illustration of our setup, H-hand setup, which is at the Bogomolets Institute of Physiology in Kyiv. And you see that green laser illumination here, and that is a HP pen, and we record single channel activity. And this is what we see through the microscope. This is a tiny smooth muscle cell here. It's a tip of HP pen. Now, what about these gold nanoparticles? They are colloidal gold nanoparticles. They have distribution of sizes where we examined, and the peak is at about 5 nanometer. And the optimal irradiation causing plasmon resonance could be worked out, and it's at 530, about 510 and 570 nanometers. We used 532 nanometer green laser. Now, again, we showed this effect at whole cell level, quite substantial. We also tested stabilizers of these gold nanoparticles, but it didn't produce. So mostly the effect was due to gold nanoparticles plus lasers. The largest effect was this. And these are actual recordings of whole cell levels. Now, again, it was very revealing to see what happens in that single channel level. Again, we record PK calcium channels in smooth muscle, expressed in smooth muscle cells, and see what happens in this case when the channel opens in control, quite occasionally, not much activity. Now we apply gold nanoparticles alone. There is some increase in channel activity, definitely there is. Here, open probability is increased a little bit. But then you turn on laser, and you can see huge effect of this gold nanoparticles, channel activation. You can reverse the order of these interventions. First, we used laser illumination. Not much is happening by laser alone, but then after we had gold nanoparticles, we observed very large increase in channel open probability. And as a biophysicist, I would like to show you and explain one very interesting effect which relates to a number of active channels. Actually, if you record from a membrane page containing several channels, then the distribution of numbers of open channels at a given moment, it follows binomial distribution. But here what we noticed, this was very strange observation, because we see what happens. We observe up to 4 channels open here. Here is simultaneously up to 4 channels open. But these 2 open very occasionally, just like in control. Apparently, these 2 have been activated very strongly. So how this is possible? Usually, pharmacological drugs, they affect equally all drug targets. So we used binomial distribution to work out probabilities, and you see in control, the probability that all channels are closed, 1 open, 2, 3, or 4 channels open. This probability follows very nicely the observed values, calculated, shown by these bars, and observed values shown by some circles. So, very good correspondence between theory and real observations. But with gold nanoparticles, we have deviation from this principle. There is no more, we don't see this follows binomial distribution. And that means that gold nanoparticles, they affect just 1 or 2 channels, not all of them. They cannot reach all of them in the membrane. And what I should have mentioned, actually we apply gold nanoparticles to the external solution, but the channel from which we record is enclosed by page pipette. So these gold nanoparticles, we can be absolutely sure, they have no direct access to the channel. So this is really remote. And we use this to see if this could be physiologically relevant. And what we showed that plasmonic gold nanoparticles can induce vaso relaxation. We used pulmonary arteries isolated from rats, reconstructed with 1 micromolar noradrenaline, and then gold nanoparticles, by activating PK calcium channels, they can counteract the action of noradrenaline by membrane hyperpolarization. And we solve this dose-dependent vasodilation in aorta and also in pulmonary artery, which you can induce simply by laser illumination. So that was really great to see physiological function, which is modulated by laser illumination in the presence of those nanoparticles. Now I'd like to move to a different area. Now let's talk, I think it's the end of our lecture, let's talk about fullerenes as blockers of two types of different ion channels. Again, beta calcium channels, with which you are already familiar, and also receptor-operated TMPCC channels. And finally, single-wall bond nanotubes, also as blockers of TMPCC channels. Now, we have done some molecular modeling work with fullerenes, and remember those papers I showed at the beginning of my lectures. Molecular modeling data, we found pretty much similar things with fullerene, including beta calcium channel 4, and that interaction is pretty much stable. And experimentally, what we found that C60 fullerenes applied at 7.5 micrograms per mean, they substantially inhibit whole cell currents in pulmonary artery myocytes, and these are mean current voltage relationships in control and in the presence of fullerenes. And here the time course of this effect. I should note this inhibition develops very slowly, maybe suggesting that the fullerenes need time to get to its binding site. And how this is possible, that was a good question, and I will talk about it. But first I would like to show you some data that illustrate some selectivity, at least some selectivity of this effect. So while we observed inhibition with low buffering of calcium, when beta calcium channels are active, if we buffered calcium very strongly in the cycle, so using 10 millimole EGD as a pipette solution, then the effect is lost. And these are purely voltage-gated potassium currents. The calcium-dependent component was inhibited by high EGD. So at least we know that C60 fullerenes, they do not interact with voltage-gated calcium-independent channels. Now, when talking about the possible molecular mechanism of this interaction, what we have done, actually that was a question raised by one of the referees of our paper. So if we go back to this molecular model, which suggests channel pore occlusion by fullerene, we thought, okay, let's have a look at single channel gating to see what happens at that level. So these are channel openings in control, and you can very well see that when channel opens, it does this many times. We call this burst of openings. So a lot of brief openings which produce such a continuous burst of openings in control. Now, after we apply fullerenes, we can see only occasional single openings of the channel. Single channel conductance was not affected completely as the same. Just occasional openings. And that actually what it means, as a biophysicist, we can interpret this very easily. The channel should open, and after it opens, the fullerene gets into the open pore of the channel. And when it gets there, it blocks potassium flow through the channel. So next openings cannot happen. Next events cannot simply happen. So fullerene is obviously open channel blocker in this case. And again, we confirm that C60 fullerenes can be efficient relaxant of pulmonary artery constrictions. So not relaxant. When vascular smooth muscle relaxes by action of acetylcholine, and part of this effect is due to beta calcium channel openings. Addition of fullerenes can quite substantially inhibit this vasal relaxation. So they can be expected to be vasoconstrictors because they block inhibitory beta calcium channels. talking about selectivity, again in ileal myocytes, we found that C60 fullerenes do not inhibit voltage-gated potassium channels, those which are not calcium dependent. But they possibly block voltage-gated calcium channels. We see this notch, that is influx of calcium via L-type calcium channels, and that disappears in the presence of fullerenes. So potentially they are blockers of voltage-gated calcium channels, but we have not looked at that in detail yet. And now the last part of my lecture is about receptor-operated TRPC4 channels. I already explained why they are called TRP, and we worked recently on two nanostructured materials, C60 fullerenes again, and single-wall carbon nanotube. And in both cases, we found that this can be efficient block of these channels. We have actually done quite a lot of work on this current. It's denoted in the literature muscarinic receptor-operated current, MICARET. And we worked on this current from late 90s, looking at the various aspects of channel regulation, which and these various aspects are shown here schematically. So, in very quickly and shortly, that is TRPC4 channel, when it's opened, it causes calcium and sodium influx in ileal myocytes. This produces membrane repolarization, resulting in the openings of voltage-gated calcium channels. There is a further influx of calcium, and that causes smooth muscle contraction. Now, this channel is regulated in a very complex manner in synergy by two muscarinic receptors. Receptor subtypes present in smooth muscle cells, M2, coupled via GIGO, most likely through direct coupling to TRPC4, and the M3 receptor. This one causes activation of GQ11 proteins, phospholipase C, and depletion of IP2 is permissive for TRPC4 openings. And these are the illustrations that actually experimentally having such gated mechanism. We don't need to apply carbachol or acetylcholine to activate muscarinic receptors. We can increase GTP gamma-S to bypass receptor activation and activate G-proteins directly. And this is how current develops in response to GTP gamma-S. We use this voltage protocol shown here. And we isolate this current using symmetrical cesium containing solution, and we buffer calcium at 100 nanomolar to avoid calcium-dependent modulation of this channel. So this is about experimental conditions. And just to show you 12 minutes of life of this channel, as shown here, you can see it opens in again kind of burst series of long openings and short closings are alternating with vice versa, short openings and long closings. And open probability changes in time, it's from very low to very high. So looks like channels also need some rest before they can be active again. And here we've done some calcium imaging on ileal myocytes, so we can appreciate the physiological effect of this activation of this current is to induce calcium rise in the site. So then this myocyte is firmly attached to glass cover slip, that's why it did not contract, but we can see here calcium wave, which is nicely spread from ends of myocytes towards its side. And this is simultaneous recordings of calcium and iron ileal myocytes. Now that is schematic diagram what happens, and I'm referring to editorial industrial enterology on our more recent findings in collaboration with the University of Surrey, and actually most of this work was done there, where at last molecular identity of this channel was confirmed to be TRPC4 with some smaller contribution of TRPC6. And in that paper, in that editorial, what was said by its fourth author in the Amputar, she said that more important, based on this study, TRPC4 and TRPC6 are potentially important clinical targets that can be utilized for modulating smooth muscle function. Now, talking about clinical targets, if used as clinical targets, obviously we need some pharmacological modulators to affect channel activity. And in fact, we developed a drug called ML204, and in my laboratory we confirmed that this is very efficient blocker of this MIT current, very efficient. Then micromolar inhibits almost completely, but that is a small molecule, which is now commercially available. Now let's go back to our main topic for today, fullerenes. Interestingly, what we found using fullerenes, we already published that these findings. As a protocol here, which I already explained to you, we use voltage steps, voltage ramp to measure kinetics of the current steady-state current voltage relationships, which are shown here. What we found, fullerenes C60 at 1 micromolar, they inhibit this current almost completely. It is very, very strongly looking at the mean data, but in this particular example, almost complete inhibition. So they are efficient blockers of this channel, TRPC4 channel. Interestingly, again, similar to PK calcium channels, this inhibition develops very, very slow. It takes many minutes for C60 fullerenes to inhibit this. And we looked at biophysical features of this inhibition, just maybe skipping some details. We examined kinetics of the inhibition, we examined voltage dependence of the inhibition, rate constants of deactivation and activation, and if we compare this data to what happens due to G protein activation, these are just the opposite to what happens due to G protein activation. So, cutting this long story short, we do believe that this slow inhibition is not due to channel or blockade, like this beta calcium channel, but it is due to G protein inhibition. And G proteins are critically important for TRPC4. So we think that fullerenes actually are incorporated in the membrane. Maybe they even cross the membrane and they inhibit G proteins which activate TRPC4 channels. That's so far our conclusion from that story. And finally, single wall carbon nanotubes. Again, we have done molecular docking studies. This is TRPC4 4 subunits forming channel 4. And these subunits are shown by the different colors here. Top view within the plane of the membrane. And again, here you can see the tiny pore for ion passage. And for single wall carbon nanotubes interaction with this protein, there are actually several possibilities, at least 3. And here I'm showing you only one of those, which is SWCNP close to channel 4. Okay, this is channel 4, basically this ion permitting the channel, and that is position of a single wall carbon nanotubes. Experimental evidence also confirmed that this interaction is functionally relevant. Again, TRPC4 currents induced by GTP gamma Ns are shown here, and we measured this in mouse ileal muscle cells by techniques. And again, you can see quite slow inhibition develops after application of single-wall carbon nanotubes. IC50 looks like it's around 10 micrograms per mil. At least at this concentration on average, the carbon by current is inhibited by 50%. And again, maybe not without going into too much biophysical detail, I can just say that this inhibition is voltage independent. It happens due to shortening of state, and it's rather consistent with an interaction occurring within the plane of the channel 4 as predicted by our in silica molecular dynamics and docking solutions. So, time to conclude from these studies, and I made sure to give about 10 minutes for your questions and result questions, and also that you don't miss a coffee break. So the conclusions, maybe I don't need to say everything here, because basically I made those conclusions during my talk. So as a big conclusion from all this, this is just a strategy for biomedical application of C60 fullerenes and single-wall carbon nanotubes for regulating vascular and visceral smooth muscle contractility and dysfunctions associated with smooth muscle contractility. At the same time, we should be probably quite careful about possible side effects of these molecules, especially applied at high concentrations. And I would like to thank many people who contributed their great efforts to this research. All extant recording studies that showed that they were done by my young colleagues working at the Bogomolets of Physiology, Daria Green and Maria Bendick, and that passed in the laboratory at the Bogomolets. And I would like to say thanks to Professor Brzezinski, one of the organizers of our meeting, for our joint work on C60 fullerenes and single-wall carbon nanotubes. Vasil Hurmach did molecular modeling studies, and I repeatedly mentioned our collaborations with Institute of Pharmacology and Toxicology, especially with my good colleague Professor Antony Solovyov in developing those liposomal drugs and also gold nanoparticles. And also with Institute of Microbiology and Genetics, again, molecular modeling was done quite a lot, and pharmaceutical company Nano. And also I would like to say thanks to several grant-given bodies, thanks to the funding we could do it with the work. And thank you all for your attention.
Thank you very much, Professor Alexander Solos. So we have now you large on the screen, and I think maybe there is a few questions. I start with one which connects to the previous talk. So how far is this from application?
Sorry, was that a question? Can I see who is asking questions? I'm sorry, but I don't hear very well.
Okay. So the question was, how far is this from application to have it as a drug?
As a drug for pharmaceutical industry, yes?
Yes.
For clinical applications.
Yeah.
Well, you know that I'm a biophysicist, but what we've done in terms of drug development is classified as preclinical study. So I believe we've done pretty well at preclinical level, but we don't have any collaborations with pharmaceutical industries that could, with companies that could potentially be interested in clinical settings. Because, you know, developing drugs requires three stages of clinical trials. We simply we cannot do that. We are not medics. We cannot do that.
Okay, I guess we have more questions. Vladimir,
Should I come or?
come closer here to the microphone, please.
Can you hear me?
Yes.
Yeah. Thank you very much. You have presented many very interesting results. I'd like to understand better an interaction of nanomaterials with channels. First concern on hydrophobic nanomaterials, or at least during the whole presentation I thought that you have used hydrophobic normal fullerenes and carbon nanotubes, but in your conclusion, the last statement was written hydrophilic. What does it mean? You have used modified materials or usual fullerenes and carbon nanotubes?
Actually, that's a very good question, because we have these nanomaterials in suspension. I believe they are somehow somewhat functionalized, the surface, if I'm not wrong, to make them more water soluble. So in papers we describe them as water soluble molecules. But I think hydrophobicity is an important factor. Also, at least in case of C60 fullerenes, one of its important biological effect, it's a good antioxidant, and we still don't know how much of that antioxidant action is involved in what we see.
Okay, because if these materials are hydrophobic, I have many other questions, but if it's not clear for me how they can come into the hydrophilic channel. But if they are modified, it's clear. And another question concerning gold nanoparticles.
Yes.
You have observed some activation of the behavior of this of the system, increase of the number of channels and so on. But probably this is just a temperature effect, because if you make elimination of a membrane in which you absorbed gold nanoparticles, it should be a production of heat, because plasmonic effect will lead to finally to the production of heat. And maybe this is just a temperature.
That effect, another good question for maybe physicists. As far as I know, plasmonic resonance is a rather mysterious thing even for physicists. What we learned from, we even approach some people in the US who understand better these kind of effects. So we believe what happens here, that with laser illumination causes this, there is a gold nanoparticle with electron cloud around it. Okay, so plasmon laser energy causes at certain wavelengths, makes it move in a resonance manner, maybe quite far away from the gold nanoparticle. And when the cloud of electrons reaches voltage sensor of ion channel, which actually has positively charged amino acid resonance, then this causes channel openings. That is our explanation at the moment, which is very difficult to test experimentally, because as I said, plasmon resonance is rather complex phenomenon. But what I showed on illustrating that only one or two channels out of 4 can be activated, that is consistent with the idea that the electron cloud reaches some proteins, but not other proteins, which are probably a little bit farther away. I'm not sure if I answered your question in full.
You have suggested another interpretation, but it's not clear.
Yeah. And what is your interpretation? I would be very interesting to hear other interpretations. But unfortunately, sound is not terribly good. Maybe it could be moved a little bit closer to the microphone.
I do not know.
But try to make
So
it
it's
short,
okay.
please.
My interpretation is an increase of temperature. Increase of the temperature of the just an increase of temperature, because if you make elimination of gold nanoparticles, it should be absorption of light. This absorption of light, this light energy finally will lead to increase of temperature. At least this effect must be. If there is direct interaction, it's more difficult for me to imagine, but of course the nature is very complex.
Yes, I got that point. It's possible, but we would have to increase temperature. We work at room temperature, okay? But if we increase temperature to 36 degrees, we can maybe get a similar degree of activation. But I don't think through laser elimination of nanoparticles you can increase temperature of solution, maybe locally.
Locally, quite near nanoparticles. If you calculate an increase of temperature of 1 volt nanoparticles after interaction with just one photon, you will get very high value, which can be at the range of maybe 100 degrees or even more. Just
So,
with one photon.
Gold nanoparticle is heated itself, yes?
Yeah.
But that heat should be transmitted to the protein. And in between gold nanoparticle and protein, we have pipette tip. So protein is actually isolated from the external solution and from gold nanoparticles by pipette. Protein is in the pipette solution.
But I thought gold nanoparticles are introduced into the system and they can be absorbed on the lipid bilayer or on the protein. No?
If it gets, but it should go in, we use the cell-attached configuration. I'm not sure if I need to show that slide again, but if you remember cell, and this is page pipette attached to cell, okay? So the gold nanoparticle should go through membrane twice in the cell, and then through plasma membrane of the cell-attached connection. And I don't think that is very long.
Okay,
Thank you.
Yeah.
We have another question from the audience. Okay, you're thirsty for coffee. Okay, thanks again, Alexander, and hopefully I have the chance to meet you next time in Kiev in person.
Absolutely, Marcus. I would be happy to see you. I'm also going to have a cup of coffee. You see my personal cup from San Francisco, St. Alex. So have a good coffee break. I will enjoy mine.
Thank you. Bye.
I hope technically the presentation was okay.
I think so.
Good and sound. I couldn't hear you very well, but hopefully you could hear me.
We could hear you all the time.
Good. Wishing you for the rest of this great school. Very excellent school. Wishing you every success.
Thank you.
Okay. Bye. Bye to Paul.
Okay, so half an hour coffee break. Who needs a test or wants a test? Okay.